Good afternoon, everyone, and welcome to another edition of our immuno-webinars. Today, we have the honor and the privilege of having Dr. Patricia Bozza presenting for us. Dr. Patricia graduated in as a medical doctor at the State University of Rio de Janeiro, after which she got her doctorate degree in cellular molecular biology at the Oswaldo Cruz Foundation under the supervision of Dr. Renato Cordeiro. After her PhD, she spent a period in Harvard as a postdoc under the Pew Latin American Fellowship. And then after coming back to Brazil, she became a researcher at the Oswaldo Cruz Institute, where she currently is now as a senior researcher. Patricia is also a 1A researcher at CNPq. She's a member of the Brazilian Academy of Sciences and the World Academy of Sciences. She received in 2022 the Latin American and Caribbean Association Award for Distinguished Women in Immunology, and she also received a fellowship of the Guggenheim Foundation in 2010, and she was also awarded as an international scholar from the Howard Hughes Medical Institute. Patricia has a very impressive and very extensive CV. She has a total of over 250 published papers with over 12,000 citations as of now. And in the past 5 years, she has published over 90 papers. She is also very prominent in formation of new students. She has over 29 postdocs graduated under her, with 7 current postdocs. She also formed 32 doctors, and she currently supervises 6 new PhD candidates and over 30, around 35 masters with over 4 current supervisions. So thank you, Patricia, and the floor is yours.
Thank you, Lucas. Let's see. So it's a great pleasure to present our work to you and to discuss the data. Let me see. And thank you, Lucas. Thank you, Chris, for the invitation. The idea today is to discuss some of our recent published data, as well as some unpublished data on the functions of lipid droplets in host immune response and metabolic adaptation to infection. I will focus my presentation in the response to bacterial infection, mostly in sepsis. So the idea is to recapitulate some of the importance of studying immunometabolism in the context of infection. And I would like to show to you that lipid droplets are a very well-conserved organelle with major roles, both in the regulation of metabolism, as well as having roles in the control of infection. And at the end, I would like to discuss with you some of the data that although lipid droplets have roles in controlling the response to infection, it can also be detrimental in terms of organ dysfunction, and that has quite an important role in sepsis. So why we study immunometabolism in host pathogen interactions? So that's quite clear that the immune response requires major changes in the metabolic process to mount an efficient immune response. And that in terms of the requirement for energy metabolism and to act for functional activation of the immune cells, and that will be important for immune cells to determine their ability to divide, differentiate, and carry out effector functions. Importantly, the metabolic status of the cell can be also be reprogrammed through the influence of the pathogen, and can also have quite an important role for the survival and replication of pathogens within the cells. And that's what I would like to discuss with you today, that it's not simple as like it can be either good or pro-host or detrimental to the host, depending on how the metabolic regulation in the cell occurs and how that will end up in terms of outcomes. So we have a quite long-term interest in studying lipid droplets, in particular, the role of lipid droplets within the immune system. And it's important to point out that lipid droplets are highly well-conserved organelles. And when we say highly well-conserved, although it may have some different functions within different organisms, but they do have roles in very different organisms. And now it's getting clear that lipid droplets do have role in terms of cell interaction in very diverse organisms. So for most of you, when you think in a lipid droplet, what comes in mind is the adipocyte. That is the cell that's represented in here. And adipocytes are quite professional cells to store lipids within the lipid droplet. So they store mostly neutrolipids and have quite an important role in regulating energy flux and energy metabolism. Over the years, it became clear that lipid droplets are important in quite diverse other cells. So here in the macrophages, we can see here that lipid droplets, although they are almost not present within non-stimulated macrophages and other immune cells, they can be rapidly induced to form according to different stimuli. But then over the years, it became clear that it goes much more diverse in terms of organisms. And lipid droplets have quite important roles, both in the regulation of the metabolism, but also in very diverse other functions in several organisms. That to say, in Drosophila, zebrafish, C. elegans, even protozoan parasites like T. cruzi can have lipid droplets induced and have roles in regulating pathogen with the interaction with the mammalian cells, as well as in bacteria. Here we can appreciate a mycobacterial that contains lipid droplets within the bacteria. And it's very important to keep in mind that lipid droplets, in all these organisms, they are very important to accumulate neutrolipids, and also they are formed like this highly hydrophobic organelle that do not have a classical double membrane to circumvent the lipid droplet. But although they do not have this typical bilayer membrane, lipid droplets have a monolayer of phospholipids with a very unique fatty acid composition. And what is quite important for their functions in the different conditions is that lipid droplets, they can contain a lipidome that varies according to different stimuli and conditions, as well as a diverse proteomic that will confer different functions for the lipid droplets. So lipid droplets, they have very dynamic functions within cells by the ability to contain these different proteins that will have functions, that will confer functions to the lipid droplet, including their function in lipid traffic and energy homeostasia. So not only lipid droplets are a source of the lipids, but that they contain the different enzymes that participate in mechanisms of regulating lipid biosynthesis, lipid remodeling, as well as controlling lipolysis. So they do have functions in regulating the flux and the homeostasia of lipid metabolism within the cell. We, over the years, and that has already quite some time ago, and a project that started in the lab of Peter Weller when, so Lucas mentioned my postdoc, so when I was a postdoc within Peter Weller's lab, we described that lipid droplets are very important organelles that regulate inflammatory mediator production within a number of immune cells, and that confers this organelle with functions in regulating the inflammatory response and to regulate the ability to induce increased amounts of lipid mediators, especially eicosanoid synthesis that occurs within this organelle. So we were able to demonstrate that lipid droplets can contain major enzymes involved in eicosanoid synthesis, including lipoxygenases and cyclooxygenases, as well as the enzymes involved in the terminal synthesis of eicosanoids. In addition to that, now it became quite clear that the functions of lipid droplets are much more broad. And indeed, lipid droplets have major roles in regulating a number of signaling functions, and they can contain a number of enzymes involved in cell signaling that include PI3 kinase, ER kinase, phospholipases, p38, among others, conferring these very dynamic functions for lipid droplets. And what I will discuss to you today, it's also that lipid droplets can act as an innate immune hub that can concentrate different proteins involving in antibacterial activity, as well as in interferon signaling. Not only lipid droplets functions are controlled by their content, but also by their communication. So lipid droplets is a highly connected organelles, and their function will be highly dependent on which organelle they are contacting. And that's an area that's gained quite an important development over the last 10 years as contact between organelles change the functions and the outcomes of the response. So here is, we can appreciate that lipid droplets, they are enmeshed in a network of cytoskeleton, and that's an important aspect that makes lipid droplets move within the cell through those connections. And you can control a lot of this changing and also interactions with other organelles by regulating this interaction of lipid droplets with the cytoskeleton. For today, we are quite interested in discuss the interactions of lipid droplets with the endocytic pathway, and lipid droplets can interact with phagosomes. And what I'm going to show to you today, that lipid droplets can also interact directly with the bacteria, and that confers functions to act upon pathogens and other intracellular organisms. Here, we also see that lipid droplets are highly connected with the mitochondria network that is here stained in green. And also, this will be very important for the functions of lipid droplets in regulating the metabolism. Now, it became clear that, again, although lipid droplets do not have a classical membrane, they are able to establish contacts with other membranes through very distinct and very defined protein-to-protein complexes that enable the exchange of macromolecules within different compartments, and that will be really important to regulate the different functions of lipid droplets. Some of those contact mechanisms are now being understood, and in mitochondria, one of the key molecules is PLIN5. So PLIN5 is the molecule that enable the anchorage of lipid droplets with mitochondria. And according to the different organelle, different pairs of molecules participate in those interactions. So over the years, we were able to demonstrate that both our group and different other groups, that lipid droplets, they can be enhanced to form by the induction with different pathogens through very well-regulated mechanisms that depends on innate immune receptors. And we were able to show that TLR receptors are quite involved in the mechanisms of lipid droplet formation. For instance, E. coli and LPS triggers lipid droplet formation in immune cells by TLR4 dependent mechanisms. T-cruzi and mycobacteria are able to trigger lipid droplets through TLR2 dependent mechanisms, and different organisms and pathogens can regulate different pathways for lipid droplet induction. But it's important to say that also lipid droplet formation and accumulation in the cell will depend on the triggering signaling, but also through lipid remodeling and biosynthesis. So we see an increased lipid biosynthesis triggered by stimulation, and that involves a number of transcription factors that regulate this lipid biosynthesis. PPAR gamma and SRBP are very well-defined transcription factors involved in these mechanisms. As well, it is regulated by the uptake of lipids. So under those interactions, different receptors can be induced and activated to increase lipid uptake, and also the accumulation of the lipid droplet will depend on the regulation of the lipolysis of the lipid droplet, and also through the beta oxidation. One of the key steps of lipid droplet formation, especially in relation to triacylglycerol synthesis, is the enzyme DGAT1. And that is a key enzyme that acts on the last step involved in triacylglycerol synthesis and accumulation in the lipid droplet. And you will see that we are going to use some of those mechanisms to block the DGAT1 function or activation to be able to block lipid droplet formation within the cell. So in terms of bacterial infections, one of our first studies was to show that indeed the lipid droplets, when stimulated with LPS, they can activate cells to induce eicosanoids and to have roles in enhanced inflammatory condition by the production of both prostaglandins and leukotrienes. And for quite some time, we were interested on the roles of lipid droplets in response to intracellular pathogens, mostly mycobacteria that are quite professional bacteria to live within the macrophage and that have very well adapted mechanisms to survive in the lipid, in the hostile environment of the macrophage. And we've shown over the years that mycobacteria, both mycobacteria bovis BCG, M. lepra, and M. tuberculosis, they are able to trigger the formation of lipid droplets, and that act as escape mechanisms for the mycobacteria. There is an association of the lipid droplet with phagosome containing bacteria. The bacteria can take up the lipids from the lipid droplets for their growth and replication. And also, they can modulate the response through lipid droplets to produce a more prone environment that that mycobacteria can survive within the macrophages with the increased production of PGE2. And for quite some time, we've established that lipid droplets for a number of intracellular pathogens, they act as a mechanism pro-pathogen that enable pathogen survival within the cells. More recently, some studies start to highlight that lipid droplets could have a role in regulating the killing of the pathogen as a very conserved innate mechanism for this organism. So one of the very interesting studies was a study in Brazil that have shown that lipid droplets are capable to armazenate histone, and through very stimuli-dependent mechanism through GABA, this histone could be delivered out of the lipid droplet and kill bacteria through this mechanism. So we've been, after that, recently quite interested to understand how was the roles of those lipid droplets in the killing of pathogens in the context of a mammalian infection. So today I'm going to focus on the sepsis model and some of the other extracellular bacteria infection to show to you that the functions of lipid droplets in host innate immune defense can be both in a way favoring the killing of the pathogen, and also to discuss with you some of those metabolic adaptation to infection and how this can contribute to the outcome of sepsis. So why we are interested in studying sepsis? So sepsis is defined as a life-threatening organ dysfunction that is caused by a dysregulation of the host inflammatory and metabolic response to infection. So although the infection is important to be present to define the sepsis, the problem in sepsis is much more the response, the inappropriate response towards the infection, sometimes even more than the pathogen itself. So globally, 20 to 30 million patients are affected every year by sepsis. It's still the most prevalent cause of death in the ICU. Each day, around 24,000 people die from sepsis worldwide. It has enormous economic cost, and the incidence is projected to keep increasing. That due to the population that is getting old and more susceptible to sepsis, as well as by the emergency of multi-resistant bacteria that can also contribute to that. In Brazil, there's over 400,000 cases per year, with over 200,000 deaths per year of sepsis. So it's interesting to think, and here, which is like a class for the PhD program, how our science goes, and not in a linear way. So when I was thinking about the preparing presentation about the sepsis, so actually my first independent study as a research back in Brazil was to study the role of lipid droplets in the innate immunity and in the sepsis context. And at that time, we were able to show that LPS induced leukocyte lipid droplet formation. That was important for leukotriene synthesis production. We demonstrated that patients with sepsis have increased lipid droplets within their peripheral leukocytes, what is shown here in this graph. And not only that, but we showed that lipid droplets were also sites for cytokine storage within sepsis. So we show that for TNF and MIF, which is a quite important cytokine in sepsis. And also we show, and that is something that we still need to explore more, that some of those cytokine receptors were also present within the lipid droplet. So, and that comes to the studies that we are in today, that we are getting back to this, and that was through a collaboration with the groups that first described that mechanism in Drosophila, that lipid droplets had an antibacterial component in Drosophila. We teamed up to try to understand if in mammalian cells, these mechanisms would also be present. So that's a collaboration with Albert Poe in Barcelona, Robert Parton in Melbourne, Stephen Grass in California, Marta Bosch also in Barcelona, and the work mostly of Felipe and Livia in my lab. And here, so we started to try to understand if those lipid droplets from mammalian cells, and here we are using hepatic-derived, liver-derived lipid droplets, would have functions to mediate the killing of bacteria. This first experiment that I'm showing, we purified lipid droplets from LPS stimulated animals. We purified the lipid droplets out of the liver, and we can use the way that lipid droplets, they are buoyant in a sucrose gradient. We extract the proteins from the lipid droplets. So in this case, we were not using the lipid component of the droplet, but the proteic component. And then we incubate the lipid droplet proteins with E. coli, and would ask if those lipid droplets would have a capacity to kill the E. coli. And here is just the purification of the lipid droplets to confirm, and we use clean 2 as a mark of lipid droplets. So we purified the lipid droplet component out of the gradient. This purified the lipid droplets then we show here that are capable of restricting E. coli growth in the plate, so to have a direct effect on the E. coli. And also, would they be able to show that indeed lipid droplets can establish direct contacts with the bacteria, and that's what we are seeing here. If you pay attention in this figure, the lipid droplets are capable of make contact with the bacteria, and also to disrupt the bacterial membrane, as you look here in detail, and we can quantify those contacts and these effects over bacteria. So we went on to try to understand what were the components within the lipid droplet that could participate in this mechanism. Obviously, the first guess was the histone, and indeed, we had previous data to show that lipid droplets in different mammalian cells can contain histones. We did an enormous amount of experiments to show that in this case, histone was not involved. So we try both by staining, by antibody blockage, in different assays, and we could not confirm that histones were involved. And then we went on to analyze by proteomics what would be the components present within the lipid droplets under these circumstances. And as you can appreciate here, lipid droplets, they make quite a hub of interferon-dependent signaling, so with an accumulation with our IRGM3 and a number of interferon-dependent pathways. One of the proteins that call up our attention was CAMP, or seen here, and also viperin, that are proteins that have peptides that have antibacterial activity. And we went on to try to understand if indeed we could confirm the presence of those proteins within the lipid droplet, and to try to understand if those proteins were really involved in regulating the killing of the bacteria. So as we can see here, if we do Western blot from purified lipid droplets, we can confirm the data from proteomics to show that CAMP, viperin, and IgTP are present within the lipid droplet fraction under the gradient. Also, by immunofluorescence assays, we can see, and also by proteins, fluorescent transfected intrafluorescent proteins, we can show that indeed viperin and CAMP are localized within the lipid droplets. And if we silence CAMP within the cell, we can see that we can block the effect of lipid droplets to do the killing of the bacterium. So we went on to see if sepsis would also trigger this capacity of generating lipid droplets with the capacity to have antibacterial activity. Here we can show that in the CLP, so the cereal ligation and puncture model of sepsis, we can see that there is a drastic. Eu consigo apagar aqui? So there's a drastic accumulation of lipid droplets in the liver of the animals within the sepsis within 24 and 48 hours after sepsis. And again, we did that same assay to see bacterial activity within the lipid droplet. So by extracting proteins from the liver, from the lipid droplets from the liver of septic animals, we can see that the lipid droplets under those conditions have a much increased capacity of killing of E. coli when compared to the control or to the sham-stimulated lipid droplets under these circumstances. Now we are trying to understand that further, what are the proteins and mechanisms and if other proteins are involved and how this is regulated in the macrophage to contribute to lipid droplet, to the roles of lipid droplets in bacterial killing. So in this assay, which was a monography of Rodrigo, and it's now a manuscript submitted for publication, we can see that if we modulate the levels of lipid droplets within the cell, that have an impact in the control of E. coli growth in or the E. coli killing within the macrophage. And that's what we see here. So if we stimulate, if we infect the cells with E. coli in the presence of oleic acid, that would enhance the number or the accumulation of lipid droplets that have a role in restricting the killing of the E. coli. So the more lipid droplets we have, the more is the killing. If you block the lipid droplet formation with the inhibitor of DGAT1, we partially restore the killing capacity of the, we can see an increase in killing under these circumstances. So we went on to see or enhance the growth of the E. coli and stop the control of the infection. So as we can observe here in the macrophage, now is the whole macrophage and not the lipid droplet itself, we can see that indeed if we inhibit the lipid droplet formation in the macrophage that were infected with E. coli, we can see a reduction in the expression by the Western blot of the antibacterial proteins that we previously demonstrated that were present within the lipid droplets. So CAMP, Viperin, and IRGM3 were all reduced when we reduced lipid droplet formation in the cell. And now going to the in vivo model, we did the same assay. So we treated the animals in the CLP model with the DGAT1 inhibitor. So to inhibit lipid droplet formation within the animal. We did these experiments without antibiotics and looked for the bacterial in the peritoneal cavity and in the plasma within 6 hours of sepsis. And as we can appreciate here, animals that were treated with the DGAT1 inhibitor, they have less lipid droplets that are being formed in the peritoneal cells, and they have an increased bacteria in the serum that can be shown here, and also an increased bacteria in the peritoneal cavity of those animals. And again, some of the same proteins that we've been shown before are also present on those in vivo formed lipid droplets triggered by the sepsis. And that come to the conclusion of this first part of the talk, that indeed mammalian cells, lipid droplets from mammalian cells, they can be formed upon bacterial infection and sepsis. They can exhibit antibacterial properties in purified lipid droplets. Also, that lipid droplets can establish contacts with direct contacts with the bacteria that would enable the delivery of the antibacterial proteins towards the bacteria. And indeed, if we look at that in vivo situation, we can modulate the killing of the bacteria by modulating the levels of lipid droplets within the cell. Importantly, just to remind you of that other part, that's not true for all bacteria that we study. So in Salmonella, for instance, lipid droplets, they are induced during infection. They are induced through highly regulated mechanisms that depend on the virulence of the bacteria. And those lipid droplets that are induced, they are involved in inflammatory mediator production, mostly PGE2, but different than E. coli, they have a role pro-pathogen and can modulate the replication of the bacteria within the cell. So then we wanted to get hold on what were the effects of lipid droplets in the regulation of the metabolism, and if they would have roles in regulate the immunometabolism in during inflammatory conditions. And in this sense, it's important to think that lipid droplet mitochondria contact plays diverse roles. So they are very important to regulate for thermoregulation, for energetic metabolism. Also, lipid droplets are conserved through evolution in a mechanism to avoid lipotoxicity. So free fatty acids, they are very reactive. They are very toxic for the cell. So to accumulate that under triacylglycerol, it's a much more inert lipid that would not damage the cell. However, it's been shown that according to the situation, lipid droplets can itself have a pro-lipotoxicity role and contribute to insulin resistance, non-alcoholic fatty liver, and also to mitochondrial dysfunction. So we went on, so we went on in that LPS stimulated model, looking at the liver to try to understand how those lipid droplets would regulate the effect on the mitochondria. And we can see here that lipid droplets in the liver can establish contacts with the mitochondria. We can quantify these contacts, and upon stimulation with LPS in the liver, we saw a decrease in the contact of lipid droplets with the mitochondria that comes together with a decrease in the beta oxidation, towards the mitochondria. And although we see an increase in PLIN2, that's one of the markers of lipid droplets, especially when they're in different cells, that we can see here by Western blot that PLIN2 is increased, we see a decrease in the expression of PLIN5, that protein that's involved in lipid droplet mitochondria contact. Those lipid droplet mitochondria contacts, they could be restored if we transfected the cells with PLIN5. So that would lead to an increased lipid droplet contact with the mitochondria. And getting back to the beginning on how lipid metabolism or metabolism as a whole is quite well connected with the immune response, we see that those mechanisms to regulating the contact and the metabolism of the cell, they do also have impacts on the killing of the bacteria within the cell. So, as a partial conclusion, our data suggests that in response to infection or danger signals, lipid droplets, they have their proteome altered, and they accumulate antimicrobial proteins. They also participate in other inflammatory pathways, and they also regulate interferon signaling within this situation. Lipid droplets in the liver, they dissociate from mitochondria and reprogram cell metabolism with reduction in beta oxidation by the cells. Lipid droplets, they do associate with bacterial wall, and they can transfer their content to the bacteria and favor the killing of the bacteria under these circumstances. So in the last part of the talk, I'd like to bring to how those lipid droplets would then participate in the sepsis itself, on the response of the animals and to the organ dysfunction that we observe in sepsis. So it's quite important to say that reprogramming of metabolism in sepsis occurrently in a very strong way, and lipid metabolism reprogram is an important aspect of this regulation. And lipid metabolism reprogramming has been associated with poor outcome in terms of death of the animals, of the patients and the animals with sepsis. Also, there is an important mechanism that regulates mitochondria within sepsis, and it's been well established that there occurs a mitochondrial dysfunction within septic patients, and that is a key mechanism in terms of the outcome of the patients, and the patients that have the more of mitochondrial dysfunction are the ones that evolve with non-survivors. So we were then interested to try to understand if lipid droplets, according to the circumstances and in sepsis in particular, could participate in a dysregulate mechanism or in a dysregulate role by contributing to a disruption in the tolerance mechanisms of the tissue, and by consequence favoring organ dysfunction and death of the animals or patients. Here I'm going to use mostly in the animal model of CLP. So in the immune response and as a defense mechanism in response to infection, there is a quite delicate balance between to resist to the pathogens or to induce killing of the pathogens, and the capacity to regulate the response of the tissues and the survival, or a mechanism that we call tissue damage control or disease tolerance, that are mechanisms that control and regulate the damage towards infection to protect tissues and favor the tolerance to infection. So as I've shown to you, lipid droplets may have roles that would regulate pathogen killing, so contributing to the mechanisms of resistance to the pathogen. So we wanted to understand what would happen in a very pro-inflammatory and dysregulated mechanism as in sepsis, and if in these circumstances lipid droplets would keep this mechanism as a way of protecting the host towards infection, or if in this case could contribute to organ dysfunction. So getting back to a similar figure that I showed to you in the other slides, here is the CLP in mouse. We can see that in Schwann, in the liver, we almost do not see lipid droplets or very low levels of lipid droplets within the liver of Schwann operated mice. However, in sepsis, and we can use different puncture and number of punctures to graduate the severity of sepsis, we can see that the lipid droplet numbers would increase according to the severity of sepsis, of the insult. And indeed, when we look for markers of organ dysfunction, so here we are looking for AST and ALT, so two enzymes that are present in the hepatocyte that would be released upon damage of the hepatocytes. Those are the transaminases that get elevated upon liver damage. We can see that during sepsis, there is an increase according to the severity of both transaminases, and that occurs and correlates with the number of lipid droplets in the cell, as well as albumin decreases upon sepsis. That's a marker of liver function. So the liver under dysfunctional properties would not be able to synthesize albumin. So we see a decrease in albumin, which also is associated with the number of lipid droplets. So we went on to try to see if indeed the lipid droplets could contribute to the organ dysfunction that we are seeing in the model of CLP. And first we did lipidomic analysis of the lipid droplets. And that's to show that indeed, as I told you in the beginning, not only the protein content change upon sepsis within the lipid droplets, so this is the lipidomic of purified lipid droplets, but also the lipid content within the lipid droplet, they also change. And interestingly, that change involves an increased amount of unsaturated fatty acids that we can appreciate better in this figure. So unsaturated fatty acids, they are again more reactive, and we can see here that arachidonic acid, they accumulate during sepsis condition, as well as a number of lipid species, especially the unsaturated lipid species, are increased during sepsis. So we went on to see if there would be an increased in lipid peroxidation occurring within the liver of those animals. And we can see here that indeed during sepsis, and here we can see not by staining the lipid droplets, but this aspect of the foam cell when you looked on the tissue that was fixed and submitted to alcohol dehydration, we can see that there is quite an increase in lipid peroxidation as measured by the increasing T-bars under this tissue. And we asked if lipid droplets could be then involved in this process of lipid peroxidation. And the answer to that is yes. So if we purified lipid droplets from sepsis, we see an increased content within the lipid droplets of a marker or of isoprotein, which is a very good marker of lipid peroxidation. That is a mediator that is formed after the non-enzymatic metabolism of arachidonic acid. So it's a mediator that would be produced by the peroxidation, non-enzymatic peroxidation of arachidonic acid. And that has been used as a marker of lipid peroxidation in different tissues. And we can see that that's quite increased in lipid droplets that were purified during sepsis. And by using in situ staining for lipid peroxidation probes, we can see that indeed we see an increased, and here we used Bodipai C11 as marker for the oxidized lipids. We can see that indeed lipid droplets are sites for increased lipid peroxidation occurring with the cell. And also if we use a marker that would stain the protein that has suffered the effects of lipid peroxidation, that's that we can see here, that lipid droplets are sites of the protein modifications upon lipid peroxidation effects on macrophages that has been stimulated with LPS plus interferon, and as a similar response in the macrophage that has been recovered from the peritoneal cavity of animals with sepsis. So we are now intrigued to try to find out if indeed the lipid droplets can, according to the inflammatory condition, contribute to mitochondrial dysfunction. And different from what we saw in the liver, in the macrophage, we do see an association of lipid droplets with dysfunctional mitochondria. So that association increases upon sepsis in the macrophage, and especially in the damaged mitochondria are in high contact with the lipid droplets. So we went on to see if, so let's use that strategy to decrease lipid droplets. Would that protect the liver from the deleterious effect of the lipid droplets? And that's what we are looking here. So using the DGAT1 inhibitor, we can appreciate with this treatment indeed dramatically reduce the accumulation of lipid droplets within the cells. And we can see here that we can quantify this reduction in the lipid droplet here. And that also have reflect on the lipids that are being accumulating with the lipid droplet. As we've shown in the first part of the talk, when we do this treatment, we see more bacteria in the serum and in the cavity of these animals, and that's what we see here. So by CFU, we see more bacteria in these animals. However, when we treat this, although we have more bacteria, we saw a decrease in the infiltration of neutrophils towards the liver. We see a decrease in LTB4 that's being produced, a decrease in gamma interferon and IL-1 beta that are being produced under these conditions. And most importantly, we see a protection in terms of the liver dysfunction when the animals were treated with the GAT inhibitor. So we see a reduction in the transaminase enzymes with an almost restoration of the ability to synthesize albumin and a quite important reduction in different markers of lipid peroxidation that are occurring in the liver. So that comes to our conclusion that under a very inflammatory and dysregulated situation, so although lipid droplets can have pro-host effects and control bacterial infection, during sepsis that you have a dysregulated pro-inflammatory condition and altered metabolic response, the lipid droplet accumulation, they associated with severity of disease and organ dysfunction. They accumulate unsaturated neutrolipids and lipoperoxides. When we inhibit lipid droplet formation, we can inhibit lipid peroxidation and restore the function of the liver, thus suggesting that lipid droplets, they can have multiple functions during bacterial infection that will depend on the bacteria as well as on the articulated host response that will lead under these circumstances of a dysregulated response. Lipid droplets may contribute to sepsis-associated organ injury and can amplify the lipid peroxidation and inflammation, thus suggesting that the lipid droplets can be used as a target to control organ dysfunction in one way. However, keeping in mind that it also has antibacterial activity, which is also another side that can be explored in terms of multi-resistance and to think how lipid droplets can contribute in these areas as well. So to conclude, pathogen, they can induce regulated biogenesis of lipid droplets, and that is important not only for sepsis, but to a numerous diseases of health, human health importance. Lipid droplets, they function as signaling platforms for inflammatory mediator production, not only for lipid mediator production, but a much more broad sense in terms of also regulating cytokine signaling. Intracellular pathogens may hijack lipid droplets for their survival and replication. That is quite clear for pathogens that are obligate intracellular pathogens, as is the case for viral infection, like for HCV, for COVID, for dengue. Lipid droplets, they favor the replication of the virus within the cell. That's also true for mycobacterial infection. However, they do have evolutionary conserved functions in host defense, and they can control the replication and induce killing of bacteria, mostly extracellular bacteria. And as I showed to you, lipid droplets can also contribute to the dysregulated metabolic response towards infection and contribute to organ dysfunction in sepsis. And to finish, I'd like to acknowledge the many contributors of this study. As I mentioned, most of the work that I showed was carried on by Felipe Dutra and Lívia Teixeira, that has the participation of different investigators in the lab, as well as the collaboration with Marcelo from here, with the lipidomics with Sayuri from the IQ USP, Rosana Mello, UFMG with Gustavo, and our international collaborations in this study, as I mentioned, mostly Albert Poe and Robert Pacton, Martha, and Steve Gross. So thank you very much for your attention.
Thank you, Patricia, for a very exciting talk. As always, we will open first the questions to the students. If there are any questions, I would ask that everybody that's watching on Zoom that's raise their hand, so it make life a little bit easier for us, so we can identify the questions. Yeah, we actually have a ready one. Great talk. It's from Douglas dos Santos. Great talk. You have shown that the protein content inside the lipid droplets is important to modulate immune response. My question is, what's the participation of lipid composition in this response? Is it possible to modulate this lipid composition due to increased bacterial killing?
So as I shown, the lipid component is also modulated. We do see that we, especially in sepsis, we see an increased accumulation of lipid peroxides as well as unsaturated fatty acids. Those unsaturated fatty acids and lipid peroxides, they can have effects on bacterial killing as well. So that's one part that we are now also trying to understand what are the contributions for that. Even leukotrienes also may contribute to the killing capacity of the macrophage, and that's another aspect that we are looking at.
Thank you. We have a question here from Lucas Bolini. Step side of front of the camera, please.
Thank you, Patricia. I think my question actually goes in the same vein of the previous question, because I think everybody was very curious about the lipid composition. Everybody was very curious about the lipid composition, and actually what I was thinking was not so much about bacterial killing, but rather about the resolution of the inflammation. So how do you think that this lipid composition is altered in a way that would maybe help? Because of course, sepsis is a very lethal model, and the inflammation is very severe. So how do you think that the lipid droplet composition is affected in a way that would hinder resolution of inflammation? How do you think we could modulate it so as to maybe help these patients recover better, maybe inducing the production of lipid pro-resolving mediators?
So if you in the lipidomic, we can see that omega-3 fatty acids do also accumulate within the lipid droplets. Christiane has already shown that lipid droplets may form resolvins depending on the stimulation. That is something that we want to explore here in terms of sepsis. So Charlie Serhan group has quite extensive studies showing that resolvin D1 and E1, they do have protective functions in sepsis while keeping the capacity to kill bacteria. So that's something that we want to explore here. We have some data with resolvins by measuring. When we treat with the GAT, we see a decrease, but it was still not significant. So we want to explore that a little bit further to try to understand the involvement of resolvins. And again, I would say that that would also depend a lot of the stimuli, the time course of the effect, and that can have different roles of the lipid droplets in the balance of more pro-inflammatory lipid mediators or more pro-resolution lipid mediators. One aspect that we for some time we wanted to explore, and we still want to explore more, is the dichotomy when you think M1 and M2. In both situations, we see increased lipid droplets. So at first we would say, oh, we see lipid droplets only in M1, but no, we see in both situations. There are some indications that they can have different functions according to the different proteins and lipids accumulating there, but that is an area that we still need to explore more.
Right. I actually have a question before I pass to Miriam. Actually, you showed that lipid droplets, when they increase, they will decrease beta oxidation, right? So in some way, they function as a sink for these fatty acids. And what's going on with the glucose metabolism? Does it play a part, these changes from favoring one pathway to obtain energy to the other? You know what's going on?
Yeah, actually, the whole metabolism cascade, they are all very well connected. So the functions of lipid droplets in mitochondrial metabolism, it's quite complex because it will depend on the enzymes and the connections that are established. So in a way, lipid droplets are a source of lipids for mitochondria function, and when you dissociate, you can disrupt some of these mechanisms. In part, what we believe from this whole bunch of data is that up to a certain point, you have some mechanisms, protective mechanisms of defense, that when you start an inflammatory reaction, you dissociate lipid droplets from mitochondria to avoid lipotoxicity for more reactive lipids and lipid peroxidation, especially when you also, in part, dysfunction of the mitochondria also triggers lipid droplet formation. So it's kind of a function that one influences the other. However, in sepsis, it kind of goes the other way. So you have the lipid droplets contributing to direct damage of the mitochondria, and that contributing to dysfunction. The other aspect, so in terms of the glycidic mechanisms and regulation, so when you have changes in or Krebs cycle, when they are non-complete, you will also release metabolites that would lead to increased lipid formation. So, but lipid droplets in a way will also regulate lactate. So it's kind of a complex mechanism, but they are indeed connected in several different steps.
Thank you, Miriam.
Patricia, thank you very much for the talk. It's always beautiful to see different work from you. Each talk is a different work, so it's amazing. But I have a couple of questions. One of them is, do you see any situation where the lipid droplets can be secreted into the tissue, or in basically work on extracellular bacteria, or do you think it's mostly a mechanism far within the cytoplasm? And do you think they have any changes on endothelial cells as well, or is mostly on the infected cells?
So we do not have direct evidence that the lipid droplets can be released. We tried to find that a lot, and we looked, although depending on the stimuli of the cell, the lipid droplet may move towards the periphery. So when they are formed, they are, and as they are formed in high close connection with the ER, they usually are formed more close to the nuclei. With the time after stimulation, they tend towards the periphery. But together with Hosanna, we looked a lot from different EM, and we never could see lipid droplets being released. That's one aspect, but there are some other groups that have reported some occasions that lipid droplets could be released, and there are some other groups that go way far to say that lipoparticles, so LDL is a form of secreted lipid droplets, and in a way they share several mechanisms in terms of release, and also in the milk production. The group that studies mammary glands, they study the release of lipid droplets, and that is really has all the proteins of lipid droplets. They do have PLIN2, and they are the way of releasing milk together with the milk fats goes in there. So in a way, it can be released, but in our context of infection and immune cells, we could never see that. So what we believe is that E. coli, when they get into the macrophage, they are not specialized in living in there. So lipid droplets play an innate mechanism to favor the killing of those bacteria that are accidentally, let's say, inside the macrophage. Very professional bacteria that are well adapted to live inside the cell. They have several ways to counter-regulate this mechanism and to use lipid droplets as a way for their survival, replication, and disguise within the cell. So with Mycobacteria lepra, we have several images that's quite impressive that you see that the mycobacteria attracts the lipid droplets towards the mycobacteria. And indeed, we can reproduce that by using beads recovered with some of the mycobacterial lipids. They have the property to attract the lipid droplets, and they fuse with the mycobacteria, and they make like a coat. And in part, they can use that to avoid the immune response by being recovered with host-derived molecules.
All right, we have another question here in chat from Emiliana Medei. Are or could be lipid droplets involved in sterile inflammation?
Yeah, definitely. So there's a lot of work on lipid droplets in sterile inflammation. So in both of immune-mediated steroid inflammation like rheumatoid arthritis. We do see lots of lipid droplets. In Crohn's disease was one of the first description that lipid droplets could store TNF was in Crohn on material from Crohn patients with Crohn disease. Also, the very well explored and well-developed data on atherosclerosis. So lipid droplets do play major roles in atherosclerosis, both in the mechanisms of lipid droplets are formed and contain lots of the cholesterol that would come to those foam macrophages. The foam macrophages in large circumstances are macrophages enriched in lipid droplets. Those foam macrophages in atherosclerosis, they are involved in increased inflammatory mediator secretion and contribute to the enhanced response in inflammation.
All right, we actually have, I guess it will be one last question, unless, are there any questions from the Zoom room? No, so Dr. Priscilla Olsen.
Hi, thank you for your talk. Always amazing. I have a question concerning what happens to the E. coli inside the cell when the lipid droplets, come close and make them lyse. Did you follow the cells to see if these cells can stay alive, or are they gonna die because these E. coli are lysing inside them? And can this be related to the tolerance that you're talking about and you saw when you kind of blocked lipid droplet formation? And in that last model, when you saw the recovery, the liver recovery, did you also see a recovery of death of the mice? I don't know if you've shown that. And one last question, like really quick, is if you use different E. coli's or if you use different cells that would give the lipid droplets, would that change the outcome in terms of controlling the E. coli inside them? The first part, when you showed liver droplets that you purified, if you use lipid droplets from other cells, or if you use liver droplets with other E. coli, not with salmonella, or with other E. coli, would that change the outcome?
Como é que era a primeira mesmo? A segunda e a terceira eu lembro.
A primeira era se a célula morria. Né?
Ah, morria, beleza. Não, se morria. Não, lembrei. So not necessarily lipid droplet formation will impact on cell death, but several of the cells, when they are undergoing a cell death, you do see lots of lipid droplets. But you can see cells that have lipid droplets and they recover, they go back to basal levels of lipid droplets and the cells keep alive. The exact experiment that you talked, we have not done. I will ask Felipe, I'm not sure, because we followed later on, but not looking the same cell, so it's hard to say. So in the terms of the culture of the cells, we followed, and the macrophage, for instance, they are quite capable to clear E. coli, so the cells keep alive, but I'm not sure if it's the same one that killed the bacteria. We would need, yeah.
Or they're gonna die anyway.
Yeah, and Miriam had asked before, and I forgot to answer that part. Those cells have bystander effects. So some of the cells that are infected, the effect can be induced in other cells, and you can see induction of lipid droplets in bystander cell as well, not only in the infected ones. The killing part, that's a very tricky question. We have done that in different times, in different conditions. We do not see protection of the killing. That's something that we are still working on. So if we do the sepsis model without antibiotics, and if we inhibit the lipid droplet, we see an increase in mortality in the first moment. Afterwards, it tends towards not to be different. But then what we have, and that's something that we think is affecting enormously the outcome, is that the animals are not being able to control thermal regulation when they are treated with the DGAT1 inhibitor. And then the temperature decreases, they could not keep thermal regulation, and although we see an improvement in the organ dysfunction, the outcome in death, we are not seeing protection. So we are still trying to figure out how to, and using different strategies to try to overcome that. And getting back to the beginning, there's the metabolic response towards infection is quite important. So you need energy, you need control also to keep the homeostasis of the tissues. So it's very important. This mechanism we are still trying to figure out. And faltou uma parte. Nem ela lembra.
No, you said that the liver cells, you got the
No, the different bacteria and the different cells. So the lipid droplets, we've used the lipid, the protein extract from the lipid droplets in different bacteria, and we see killing also in Klebsiella, in different strain of E. coli. We do not see in Salmonella.
It has to be like drops from liver cells. If you get it then from other tissues, it wouldn't work, or would it?
I would think it would, but it's not that trivial. So we are, so I hypothesize that macrophage lipid droplet would do a similar effect because we can see the antimicrobial proteins there. But you need lots of macrophages to do this purification, to be able to do these assays. And they will be done sometime.
All right. Once again, thank you very much, Patricia, for being here with us, sharing these very exciting data. Thank you, everybody in the audience today, here and online. I'll see you guys next week. I just want to remember everybody that today is the last day for the reduced price for registration of the 5th edition of the workshop on inflammation that will happen here in Rio, October 31st and November 1st. So if you guys would want to join us at the workshop, today is the day to register. So thank you again and see you next week. Bye-bye.